Good morning, students. We have been discussing about clinical trials from the previous classes. So, in previous class, we have seen what is a clinical trial and what are the different phases. We have just seen the introduction, and we have seen just a overall of drug development, and we have seen all these things about the histories. We have seen and about the changes after Second World War in clinical research. We have seen what the problems happened, and we have seen the basic principles of drug to be in clinical trials. We have seen and types like of intervention groups and control groups. We have seen and basic considerations which you have to follow when you are doing clinical trials. We have seen and yeah, designs which we have to factors we have to consider before we choose it, trial designs and appropriateness of the clinical trials and types of clinical trials we have seen like treatment, prevention, diagnostic, screening, quality of life, overall picture of clinical trials, just a draft copy of it. And we have seen supports we need and the phases, abbreviations, and we have seen this a brief of 4 phases. And phase 1 also we have completed with the objectives of safe dose, tolerability, PK/PD of the drug, right? And we have seen prerequisites for the trials and how to conduct the trials and who are the players and what is the healthy subject according to WHO and FDA and CDSCO. And we have seen the procedures for it, how to conduct a phase 1 trials. We have seen this and designing of phase 1 trials. We have seen the designs and we have seen the estimation of dose in humans, and we have seen novel approach and novel approach we have seen, right? And next we have seen dose escalation or dose ranging and dose extrapolation. We have seen SAD analysis and MAD analysis, single dose assessment studies and multiple dose assessment studies. We have seen the pressure of single dose assessment studies and pressure of multiple dose assessment studies. We have seen and pharmacokinetics thing we have seen. Pharmacodynamic studies we have seen. Outcomes of phase 1 we have seen, right? So chance advantages. Till now we have seen in previous class, right? And now we are going to enter into phase 2. So before going into detail about phase 2, so the major objectives of phase 2 trials is to see the safe dose in patients. See, what I'm saying is, see, in phase 1, we have seen a safe dose in healthy volunteers as they don't have any disease conditions related to your drug. Okay, so there won't be much areas on side effects. But in phase 2, you are going to take a diseased people, means you are going to take the patients, not healthy volunteers. Obviously, the dose calculations will be varied when you give a dose in healthy person and a diseased patient. So we are going to see the safe dose in diseased patient. Along with it, we are going to see therapeutic efficacy. We are going to see the therapeutic efficacy in diseased people. Safe dose, therapeutic efficacy, and ADRs in diseased patients. Because in healthy patients, there are less chance to give certain drug interactions and all. And in phase 2, when you speaking in terms of patients, definitely there is a high chance of showing certain high ADRs and all. So that we have to consider. We have to need to have another batch of it here, right? Okay, going into phase 2. The phase 2 is called as a therapeutic exploratory phase. So why this one is being as a therapeutic exploratory phase means this is the first time in clinical trials we are seeing the efficacy or therapeutic effectiveness of a drug in humans. Right, in preclinical trials, we have used animals and we have seen therapeutic efficacy in animals. But after preclinical trials, this is the first time we are going to actively and having a primary objectives of seeing the drug's therapeutic efficacy. So that's why we call this one as a therapeutic exploratory phase, right? So in phase 1, we already done with safety of the drug, right? Then this phase is to see the effectiveness of the drug molecule. Okay, and we'll be doing this study in a group of tens to hundreds, means from 50 to 300 and 50 to 500 or 50 to 800, like that. Okay, And here also we have failure rates in phase 2. So if the failure in phase 2 is because of the drug candidate, as the drug candidate won't work as intended, are it showing toxic efficacy? Means, see, as we see in phase 1, we just see the safe dose, we don't see therapeutic efficacy. So it might be that the drug has worked appropriately in animals, and in humans also they found a safe dose, but when it comes to therapeutic efficacy, whether to show its intended activity or not, the drug may be lagging certain importance in it, where the drug is safe, but the drug don't have any therapeutic efficacy to use. So at that time, the drug is going to get rejected from phase 2 trials. And apart from that, if the drug is showing certain toxic levels, so why I am saying toxic levels is because when we see in phase 2, phase 1, we have seen the healthy volunteers. So there might be a proper absorption, metabolism, elimination, or what you call as metabolism and elimination in healthy volunteers. But when you see a diseased person, when you see when you take patients in phase 2, obviously the systems or their system of health might be reduced when compared to healthy volunteers. So at that time there is a high chance of sedimentation or improper metabolism, which improper detoxification, which leads to sedimentation of the drug in the body, and which leads to certain side effects or toxic effects. Yeah, if the failure is because of toxic effects, the drug is going to abandon. Means, so these drugs you are going to keep in a compound library. You are going to keep in a compound library, and you'll stop the trials. Okay. So as you know, once we keep the drug in compound library, what we are going to do with it, right? Next, if the failure of the drug is because of the efficacy, means the drug is not showing any toxic effect, but there is a problem with showing efficacy. Means there was no, supposing if your drug is for stage 2 or stage 3 of cancer, but if drug is not showing that much appropriate thing to see to treat the severe cases or final stage of cancer, then it's not an up to mark, right? So what you will at that time, you will stop the trials. You don't terminate it, you will stop the trials. You will go for further analysis. It will be done to check unfavorable effects. What was the reason why the drug was not showing a therapeutic efficacy at a higher rate, where we have to treat the final stages or higher stages of any disease condition. You will see it, you will keep the drug with you, you will do any modifications required. If there is any modification required, you will do the modification, then you will start the trials once again. Okay. And about one third of experimental drugs successfully complete phase 1 and phase 2 trials. There won't be any problems much, only one third. Remaining two thirds will be having one or other issues, maybe toxic effects or not working properly, not showing the highest efficacy which is needed, and about one third of the experimental drugs are successfully completed. So, yeah. The next one is, yeah. So early phase 2 trials uses doses that has been observed to be safe in phase 1. So whenever we dose in phase 1, we'll be dosing based upon the data what we got from preclinical studies, right? We do dose escalation, dose extrapolating, and we'll do it. When it comes to phase 2, we'll be considering the data from preclinical trials and phase 1 both. Okay, we'll be considering the dose from preclinical trials and phase 1. Then we start giving dosing, and the first dose given in phase 2 trials is the dose which is observed to be safe in phase 1 trials. Okay, so that dose we will be seeing to see the pharmacological effect of the drug, to see the pharmacological effect of the drug. Okay, and it is done to establish if the drug is having a therapeutically equal interventions, like if the drug is therapeutically useful in intervening, okay, any disease conditions. Okay, like if you see this point to establish if this is a therapeutically useful intervention or not, therapeutic intervention or not. Why we are saying therapeutically useful or not means, if there is already existing drug in the market, for the disease condition, and you are producing a new drug for the same disease condition with higher benefits. You thought of higher benefits, but when you see the comparison between the existing treatment in the market and your drug, and if you don't find much difference between these two drugs, then obviously your drug is not going to get approval. Even if you complete the phase 2 trials, for getting phase 3 approval, you have to submit the data. So at that time, the government or the agencies like CDSC, ODC in India may stop it, as your drug is not having any special approaches or novel approaches or benefits over existing treatment in the market. Generally, these things will be seen with only a single dose of the drug. Like in phase 1, we have seen single dose and multiple dose ascending studies. So we'll be dealing with only single dose drugs. So later in phase 2 trials are conducted in patients to establish a safe dose regimen. Like this one is in phase 1. This is phase 2. First time we are doing in animal. We are doing in what we call as humans. So we'll be just checking on it to see the safe dose. So first time we already seen the safe dose in humans in phase 1. But the problem what was happened there, the problem was that it was just in a patient, so there won't be much, there will be less chance of adverse reactions or anything. But in here we are dealing with patients, definitely there is a high chance of any ADRs or side effects, right? Next, what are the objectives, right? First one is to explore the therapeutic efficacy and safety of the new medicinal product. So as we see, this is the first time we are seeing therapeutic efficacy, right? And safety of the new experimental product. And next one, aim to identify the side effects, most common side effects associated with the new medicinal product. See, the side effects and adverse drug reactions can happen at any dose. Can happen at any dose at any phase. It's not that side effects should be happen only in phase 1 or in therapeutic efficacy should be seen only in phase 2. Or confirmation of the thing should be happen only in phase 3. So what happens when we see the series of cycles? So it's just like a mixed thing. Like we say, in phase 1, if you see the objectives of phase 1, so you will be seeing the safety, tolerability, PK/PD, right? So when you're dealing with single dose ascending or multiple dose ascending studies, at one point of time, you are increasing the dose. At that time of increasing the dose, definitely you might be seeing the therapy, you might be giving the therapeutic dose. So at the time when you give the therapeutic dose, definitely certain minimal therapeutic activity of the drug has been seen there actually, right? Means you got the therapeutic efficacy dose there only, but we don't give that one much priority because our priority was safety of the drug. We'll keep the data, we'll record the data of it, but we don't give much priority to it. Same thing in phase 2, we'll be seeing the side effects, but the problem here is the side effects was not the first major objective of the phase 2. The major objective is therapeutic efficacy. But why we are dealing with these side effects is because this is the first time the drug has been given in a patients, right? First time the drug has been given in a patients. Next one, risk-benefit assessment means the objective of this phase 1 on this one is to provide essential risk-benefit assessment before more new patients are exposed to the new drug. Like we see even in phase 1, I said we'll be doing in a batches, batch 1, batch 2, batch 3. So batch 1 will be done with the studies, right? So if there is no problem, batch 2 they will do the higher dose, and in batch 3 the next higher dose if there is no problem. Like that, so here we are going to see the risk-benefit ratio, risk-benefit assessment. Like your best example is your anti-cancer drugs, right? So when you use anti-cancer drugs, obviously you have certain side effects. But why we are giving instead of even having the side effects or ADS mean the benefits are higher here than the risk. That assessment will be done. Next, the secondary or additional objectives are evaluation of potential endpoints. Means what is the end dose? What is the maximum dose can be given to the patient to show the therapeutic efficacy? Can be seen, should be seen here. Next, therapeutic regimens, including concomitant medication, means along with a specific drug, if patients having other comorbidity conditions, which can be given. Like supposing if your drug is for hypertension, Okay, a patient may be having other complications like kidney problem or liver problem or some other problem. So you'll be seeing any specific therapeutic regimens. Or other example, for your peptic ulcer disease unit therapeutics, you'll be having 2 drug regimen or 3 drug regimen. And for COVID nowadays, we have drug regimens like HCQ, azithromycin, pantoprazole or ranitidine, and paracetamol and vitamin D3 and calcium supplements and multivitamin supplements. So all these are regimens. So those kind of therapeutic regimens will be seen in as the major objective of your phase 2 trials, right? Next one is target population, mild to severe illnesses for further subjects in phase 2 and phase 3. So here you have to target which group of people with mild to severe illnesses should be studied for phase 2 and phase 3. Next, prerequisites for phase 2. In phase 1, the prerequisites were data from preclinical trials and approval from the government agencies. Okay. Next one, just like that, your prerequisite for phase 2 is animal toxicity studies, which need to be which has completed before phase 2, and the data from phase 1. These are the these are the two prerequisites we need before we start phase 2, like the toxicity studies, like repeated dose toxicity studies in animals in 2 different species. For a time equivalent to length of phase 2 studies means if you are going to, like we seen, if you are using a drug in humans for 1 month, the drug test, toxicity test in animal should be done it for 3 months or more than 3 months. And if you are using a drug in animals and in humans for 3 months, that drug should be used in animals for 6 months. So like this, there should be an equal and improved length, almost equivalent lengths which are needed for the phase 2 of clinical trials. Okay. Next, pharmacokinetic studies. We should have a data pharmacokinetic studies, mutagenicity studies, like in both in vitro and in vivo studies are needed to do it, right? So in vitro is nothing but in compound models, and in vivo means in living things. The next prerequisites are early phase clinical trials data, like outcomes of phase 1 studies, where we get a preliminary safe dose, like when we see we can give maximum of 1 gram of paracetamol per day, means not per day, per time. 4 grams per day is maximum safe. So those data will be getting, and which is nothing but your tolerable and adverse drug events dose that should be taken care. That is prerequisite for it. And next, obviously, the regulatory authorities and ethical committee approvals, which plays a major role. Like because why I'm saying is, after completing each and every step, you are going to check. You are going sorry, not you are going to check. You are going to submit the application to the CDSCO or DCJ in India, right? So obviously they're going to check the plus and minus which are happening in previous phase, and based upon that, how you are going to plan this next phase, or do you really have the data which will sufficiently support you for next phase? Suppose thing. So after phase 1, if you find out there is a more toxic effects, and if you still you want to continue, you don't be getting approval from the government agencies or CDS or DCA, because they have seen your data and find out there is unnecessary risk which are happening in patients when you are doing it. And there is no much, like in phase 1 you will be seeing a little bit of toxic, little bit of therapeutic efficacy, and that was also nil at that time. Then obviously there is a less chance of getting approval and more chance of rejecting from the trials. Okay. Next, type of phase 2 trials. So like when we see the major approach or major objective of phase 2 trials was to get the objective, sorry, not the objective, to get the therapeutic efficacy of the trials. So here we have to consider two things, right? First one is how much drug should be given to see the efficacy, and how the drug is being showing its effects when you certain dose. So like that, your phase 2 has been divided into two phases again, means 2A and 2B. So 2A will be specifically speaking about how much drug should be given to see a specific action in the body, to see specific efficacy. Let it be therapeutic efficacy first time in patients, or let it be a therapeutic regimen in patients, or let it be a safety dose or side effects in first time in patients. It can be anything. How much drug should be given to show a therapeutic efficacy in a patient. Next 2B is how well the drug works at a prescribed dose, means how well the drug works in a given dose. Like if you give, take an example of paracetamol, like if you give 325 mg of paracetamol alone to a patient who is having fever, it won't show much efficacy which was intended. But when you give 650 mg of paracetamol, obviously as the dose is high and appropriate dose, it will be showing a higher effect. So that's why your phase is divided into 2A and 2B. How much drug should be given to show an effect? It can be side effects or therapeutic efficacy. And 2B will be speaking in terms of how well the drug works at a given dose. Like when you say aspirin, the low dose will act for pain, and somewhat higher dose will act for antiplatelet, and somewhat dose change in the dose it will show certain other effect. So like that, you have to see what effect the drug shows at a given dose in a humans. Okay, this is about types of phase 2. So in details we will see now about phase 2. I mean like 2A and 2B we will see now. Right, conduct and players in phase 2. Obviously the players in conduct and players in phase 2. First one is just site. Site is nothing but a place where we will conduct the trials. That one is you have already seen in phase 1. You will be conducting a specialized hospital units, and where you should have a clear, closely monitored trained investigators. Because in phase 1 we have used patients, no much effects, no much precautions to be taken. Like we should take precautions, but we should take certain higher precautions when we do phase 2, because in phase 2 we are taking patients and we are giving a new drug where we don't know exact 100% of the drug. So at that time, obviously there may be certain side effects, and there is a high chance of side effects or ADRs or adverse drug events or moments, and it can be anything can be happen. So that's why you should have a closely monitored investigators for the entire time, 24 into 7 and 365 of for taking care of the patients when phase 2 is being happening. Next one is there should be standard facility to handle an emergency or serious unexpected adverse reactions at the site. Means you should have a standard facilities to look into or to handle if there are any emergency or unknown or unexpected areas happen. Because the ADR can happen in a patient or it cannot be happen in a patient, right? It's not that this ADR is see if it is side effects, yes, there is a chance of happening, higher chance. But when you see ADR can happen or cannot happen, because it is, if you see the types of ADRs, type A and type B. So type A we can predict. Type B we cannot predict. It's bizarre and we cannot. It's not a dose dependent and we cannot predict it. So sometimes it may happen in some patients and sometimes it may not happen. Even same patients can experience sometimes and same patients cannot even experience it. Okay, so that's why should be ready to handle any emergency or serious conditions or adverse events at the site of this one, right? Next one, the medical and paramedical staff should be skilled and qualified enough to handle any emergency conditions like anaphylactic reactions, anaphylactoid reactions, any life-threatening severe conditions, or if patient went to coma in between because of the XYZ reasons. Because he's already a diseased person, you're giving a new drug which you don't know 100% about it. Definitely there may be a problem. So that's why staff should be specifically trained for basic life support things like doing ECG and all, okay, and by giving artificial breathing and all, right? So these things should be taken care properly. Next, prerequisites for phase 2. So what are the prerequisites for phase 2? Like this one, early date of clinical trial should be taken care. Okay, we already seen this. Okay, like distinctive features of 2A, like we said 2A, where phase 2 is of two things, 2A and 2B. 2A will explain, we have to see in 2A how much drug should be given to show a specific effect, and 2B how well the drug works at a given dose. 350 mg of paracetamol or 650 mg of paracetamol will act differently with that one. So the distinctive features of 2A, how it differs from 2B is 2A will be conducted at a single site, means it only one site will be conducting a 2A, it only one working place. Okay, the primary goal is to determine the therapeutic efficacy. Yeah, obviously that's the goal of phase 2 trials. And it's a pilot trials means here you don't take hundreds of people initially. You'll be starting with a less number of group like 50 or 60 people. You just do certain pilot trials. Like I already explained you what is pilot trials in previous class. You'll be doing it. Then when you do pilot trials, you'll be getting dose response determination, like how much drug should be given to show a response. The response can be a side effect either it can be anything. Next, determine dose regimens and determine of target population. This is the major mode of 2A. Okay. And next one, few hundreds of homogeneous group of patients. Means the homogeneous group of patient means the age group between 5 years. Okay, suppose if you're correct for 2A, if you're taking patients of different age groups, that will be a problem. So what you have to do is homogeneous group of people means 20 to 25, 25 to 30, or 35 to 40. That's the age group. All male patients between 25 to 30 years of age or 20 to 25 years of age, all male patient belongs to a specific colony or specific area, like in 100 meters of area or 2 meters or 2 diameters of area, wherever you are seeing. So, or you are specifically restricting it to only one community. So it should be homogeneous group, because the first time we have to see the dose response here. Okay, patients fit narrow eligible criteria. Like we said, criteria of 20 to 25 years age, all male patients belong to only one community or who are present in a specific colony or area. Okay. Next, trial design. So it will be a comparison with the baseline status, like before giving the drug and after giving the drug, you will be seeing the parameters. Next, open level, single and double blind. So as we already know, single blind and double blind. Single blind means only patient don't know what medication he is taking, whether he is taking test drug or a control placebo. And phase 2 is a double blind, means the patient also don't know and the person who is giving the drug is also don't know. So generally when you see person who is giving the drug, okay, it can be a clinical investigator. So generally being a clinical investigator or clinical pharmacologist, being a single person, it may be difficult for the only one person to control and handle everything, giving dose, collecting blood samples. So what he will be doing, he'll be having co-investigators or sub-clinical investigators, where the major investigator will instruct them, the co and sub-investigator, how to do, how much dose should be dispensed or given to other patient, other people, so that they'll be checking it. So when those people who is a co and sub-investigator, those people also don't know what they are giving it to the patient, whether it is a test or a standard treatment, a control treatment. So that is called as a double blind. Okay. Next one is dose escalation. Means once you are done with a limited number of people, then you are going to extrapolate the dose. But the first dose given in phase 2A is whichever dose was proved to be safe in phase 1. That is the first time, and then you will increase the dose. Okay. Next one, we will use a parallel dose response. Means like we already seen, one group will be receiving only the standard drug, and another group will be a standard or test drug. One group will receive standard or test drug, another group will receive only the placebo. Means the control will receive only the placebo or a standard existing treatment. So why we are giving standard existing treatment here, that we'll be discussing in further slides. Okay. Next is 2B, right? When you see 2A, it was a pilot and connected single site, but 2B was a pivotal, which was a big and a multiple sites you are going to do it. Next, objectives focus on same as a 2A, and we'll be following the blinding studies. Okay, we'll use placebo or other concurrent medicament or inactive treatment for a control group, and test group will be having a normal active your new drug, and this will be placebo or concurrent drug. Okay, next one is crossover treatments, and will be, yeah, it's multiple centers and we are going to use crossover treatments. Okay, and subjects conducted on heterogeneous population. Like in we'll stop it here for now. We'll discuss in next class.